So yeah, if you're watching this recording, I apologize. I will post a summary of chapter 5's content from another video, but the rest of this video will be from a current recording, okay? So we're starting off talking about our viral cycles. And first step is adsorption. This is when the spike binds to the surface of the host cell, all right? And this is literally just that initial binding. That's the initial adsorption. To absorb something is like soak up a liquid. That's not what we're saying. Adsorption with a D, okay? So adsorption. Number 2, if our key on our viral spike unlocks the lock on the cell surface and the cell is going to let us in, that process is called penetration. This either happens through fusing with the membrane, with the host cell membrane, or endocytosis. So that's how we get in. The next step is uncoating. This is removing the capsid and an envelope, if you have one, to release the genome. This usually involves things like digestive enzymes inside of the cell, or just binding with certain molecules in the cell can just cause the capsid to release from the genome. It just depends on the virus. So that's the uncoating. The next step is the synthesis. This means we're making genome copies as well as our capsid and virus parts. Through transcription or translation or whatever we need to be doing with our virus and everything like that. So we're making copies of our genome, but also making all those protein components and parts and things like that. Once we do that and we can assemble everything, it's our 5th step is assembly. We'll make our actual full virions. A virion is a word for a single active virus particle. Okay. This is taking our genomes, building the capsomeres, which are the parts of the capsid, building the capsomeres together to make the capsid around the genome. And then eventually we will release. That's our 6th step. Release your mature virion particles, either through exocytosis, again, just the opposite of endocytosis. When the cell puts a vesicle around you and just secretes you out, that's exocytosis. If you're budding, if you're pushing through the membrane, then that's another process that you could do to get out. So exocytosis or budding, typical ways that we're going to be doing that. So that's our general viral life cycle steps. So I apologize for not recording that whole thing because it always slips my mind when I don't start recording right away. All right, cytopathic effects. Basically, if I have a cell, this is your cell, and I'm healthy and I've got all my locks on my surface and I'm interacting with the correct keys on my neighbors and things like that. We've got all of our locks. Here's another lock. It binds with something else. Here's another lock. It binds with a triangle thing and whatever. All of your different locks on your surface, and you normally interact with normal things on the outside and whatever. And that's normal functional cell, okay? When you're infected with a virus, you're not making those. You can't adhere to surfaces anymore. You can't communicate with neighboring cells anymore, and all this other stuff. So that can lead to cytopathic effects. Cyto is cell, pathy is disease. These are effects of the cell as a result of the viral infection. And these effects of the cell that we see caused by the viral infection can be seen with a microscope. Even if we can't actually see the viruses, we can see the effects of the viruses on the cell. This would include things like when the cells are starting to get a rounder shape, rounding of the cells, especially if we're thinking about like muscle cells. They're not supposed to be round. They're supposed to be long and elongated and kind of stretched out or whatever. We don't want to see them rounding up. Even skin cells, they shouldn't be rounding up. They're nice and flat. that would be an effect of cytopathic effects, okay? Cytopathic changes. And other ones that we might see, loss of adhesion. The cells aren't sticking to each other or the tissues anymore. That can be an effect because, again, none of your locks, none of your normal attachment molecules and stuff are on the surface of your cell anymore because it's being replaced by the viral spikes. That can be a huge problem. And also, the third one is syncytia. I can never. Syncytia. A single one is called a syncytium. Okay, and what these are is, again, if I'm a cell studded with keys to the neighboring cell, which has normal locks on it, then this can cause those cells to kind of merge together. They got their own nuclei in them. This right here is a syncytium. Yes, okay. Syncytium. Multiple is called syncytia. So you can obviously have multiple cells that can kind of merge together nearby one another as a result of this. This is a common effect that we can see with certain types of viruses. One very famously known one that does this is called respiratory syncytial virus, RSV. Yes, that RSV that causes colds in adults, but very severe respiratory illness in young kids, as well as immunocompromised folks, like older folks. Okay. Those are cytopathic effects. Next, the bacteriophages. And pretty much the rest of this is kind of just keeping reference with our bacteriophages. So bacteriophages are viruses that infect bacteria. They can have two major life cycles. Some phages will just go through the lytic phase. This is whenever they are making copies of the virus that burst out of the host cell. We also have some that can switch between lytic and lysogenic. In the lysogenic phase, we have inserted the virus, the phage DNA. This creates a structure that we call a prophage into the host bacterial genome. Yes, literally in its DNA. And so again, if you're a bacteria and here's your cell, and here's your DNA, and we inserted that virus DNA in there, and that is called the prophage. Literally, that structure is called the prophage. This state, this phase of being inserted into the genome, this is called lysogenic phase. If you are no longer inserted in the genome and you're now making virus copies instead, that's the lytic phase. Any phage that can switch between these two, come on, why are you not letting me? If you can switch, you are called a temperate phage. Okay. So that's the gist of kind of lysogeny. Again, lysogenic phase is also known as lysogeny. Okay. And the way I remember this is lysogeny has gene in it. If it's part of the genes now, then if it's a prophage. And that's when the lysogenic phase is happening, okay? Okay, sometimes whenever that prophage comes back out, let me draw it for us so that we don't get lost in this. If that prophage comes back out to be lytic again, and we take our gene out and we bring with us some of that host bacteria's gene, and we package it up into our little phage structure, and then it goes and infects a new bacterial cell. And it infects it. And it can bring with it that gene. If it brings new traits with it, lysogeny is whenever we incorporate the prophage into the host bacterial DNA. Lysogenic conversion, okay? Lysogenic conversion is when you get new traits from your phage going into lysogeny. So lysogeny is when you're in your genome. Lysogenic conversion is when you get new traits from lysogeny. New genes and new traits. This can increase virulence, which means that the bacteria now causes a worse disease because of the traits it got from the phage, from the lysogenic conversion. The process of having a phage infect a bacteria and become lysogenic and getting new traits. Again, the new traits being lysogenic conversion, which is a new subset of lysogeny. This whole process is called transduction. All right. We can have, in eukaryotes, we can have viruses insert themselves in our DNA too. If it is bacterial, bacterial viral DNA insertion, we call that a prophage. In eukaryotes, we call that a provirus. Similar process, basically lysogenic in eukaryotes leads to the provirus. It can change our DNA too. If it disrupts our cell cycle enough, it can disrupt our cells and cause them to multiply out of control. If they multiply out of control, that's what can lead to cancer. Oncoviruses are viruses that go through this provirus situation that lead to cancer. They can have other genes that can actually trigger cancer production, or they can just insert in our genome and disrupt our own cell cycle. The ones that can cause cancer, though, they're called oncoviruses, just like oncology, oncoviruses, okay? All right, so that's it for our viruses. The last bit was about prions. Prions are just misfolded proteins. They are normal proteins in your body. They're normal proteins in your body. And I just want to clear this up. It takes 10 to 30 years for you to have signs or symptoms of a prion disease like Kuru or like mad cow disease. Not anywhere like a virus or a bacterial infection. It's not days, it's not minutes, it's not hours, it's not weeks, decades. No signs or symptoms at all, ever, for anybody, for decades. And it is 100% fatal. You do not survive prion diseases. Nobody does. Also, if I suspect that somebody has a viral, or excuse me, if I suspect that somebody has a prion disease caused from misfolded proteins, and they're displaying signs of dementia and neurodegenerative effects and effects of having neurological decay from prion diseases, then I have to send those samples off for testing to special laboratories, and it will take probably a couple of weeks minimum to get any result back, if you do. And they're technically not definitive when you do that. The only true definitive diagnostic test that definitely proves you have a prion disease is when I take your brain tissue out and you're dead, and I send that to the lab and look at that. The other ones are kind of like screening tests. They are indicative that you might have the disease. So if you ever hear anybody saying, oh, I went to the hospital and I got tested and I was positive for Kuru, that is a lie. Just saying. Okay. Just saying. Okay. All right. Next. All right. Spongiform encephalopathies. All prion diseases, all of them, only cause spongiform encephalopathies. We haven't seen expression of prion diseases in any other way. Can you have other signs and symptoms from that? Yes. Dementia and that type of neurodegenerative disease is how we see the spongiform effects going. But these are misfolded proteins that build up in the brain and cause holes to form in the brain from the misfolded proteins building up. Again, their hydrophobic regions are not hidden inside of the protein where they're supposed to be. So instead, they're outside of the protein and they don't interact well with water anymore. So they stick together, clump up in the brain, cause holes to form in the brain. And then that is what leads to the pathy, the disease in the brain encephalo. Okay. Just to clear that up. All right, let's go for our most commonly missed topics. Just got to check on the time. I've got a meeting in 30 minutes. Most commonly missed topics. Our first one for chapter 5. We have the organelles. So be sure you know your major organelles. These are definitely our most important ones. The nucleus, the endoplasmic reticulum, and the Golgi. Then the mitochondria and the chloroplast. These are our 5 biggies. You definitely need to know these organelles and what their functions are. They are outlined here, but I did already talk about them previously. For those of you who are watching this recording, I did already talk about them previously, I promise. But just really quick, the nucleus contains your genetic information and the blueprints for anything your cell might want to build. The endoplasmic reticulum builds your macromolecules, including your proteins in the rough endoplasmic reticulum and other macromolecules in the smooth endoplasmic reticulum. The Golgi apparatus is our shipping warehouse. It basically packages and ships things around and outside of the cell. The mitochondria is your powerhouse. It generates ATP specifically. And chloroplasts are generally organelles that are where photosynthesis occurs. We've got activation of molecules by light, so we can make ATP, and then that ATP can be used to make glucose and other organic molecules for our photosynthetic organisms, which are plants and algae. Okay. All right. In a laboratory experiment, a eukaryotic cell is exposed to a chemical that disrupts the integrity of the mitochondrial membrane. After exposure, levels and increased. Oh, I just skipped a sentence. After exposure, the cell shows a significant drop in ATP. Okay, so we're affecting the mitochondria, which is mitochondria make ATP. Now the cell has drop in ATP and increased oxidative stress. Don't worry about that too much, okay? Which of the following cellular functions is most likely impaired as a result of this chemical treatment? This is basically just asking us, what does mitochondria do? Okay, this is what the question is. Don't read too much into it too much. That is literally what this question is asking. Mitochondria is disrupted. What happens on the mitochondrial membrane? What's being disrupted? Is it protein folding and modification in the endoplasmic reticulum? No, that's not what happens in the mitochondria. Is it intracellular digestion of macromolecules by lysosomes? No, lysosomes are a separate organelle that have their own thing. That is what lysosomes do, but that's not what's happening here. Energy production through aerobic respiration in the mitochondria, of course. And then synthesis of lipids and steroids on the smooth endoplasmic reticulum. That's not what's happening. So this is actually a very wordy question that is very unnecessarily wordy, and it's actually extremely easy of a question. We messed up the mitochondria. What was affected? The answer with mitochondria in it. It's a really easy question that's just got a lot of words. Being able to read through that stuff and make sense of it is a tricky thing for a lot of people. So if you're having issues with that, you need to go through questions like this to see if you can pick out the relevant information. Our next question says a research team is studying a eukaryotic cell. Remember already, we're thinking if it's eukaryotic, it's got a nucleus and organelles. It shows a decrease in its ability to synthesize proteins. Where do we synthesize proteins? The rough endoplasmic reticulum. It's rough because it's studded with ribosomes. Ribosomes translate RNA and make protein as a product. Okay. They observe that the cell's rough, go figure, the rough endoplasmic reticulum appears damaged, and its ribosomal RNA levels, again, ribosomes are made up of ribosomal RNA, were low. So again, our rough ER is low in ribosomes. We're having a problem with translation, with protein production. Which of the following is most accurate regarding the impact on cellular functions? We should have a decrease in protein. That's what I'm anticipating. Decreased protein. I haven't read my choices yet. That's what I'm anticipating. Do we have a decrease in protein synthesis due to a malfunction? So this first part, do we have a decrease in protein synthesis? Yes, but you better read the whole choice. Do we have a decrease in protein synthesis due to a malfunction in the Golgi apparatus, which is responsible for protein modification and packaging? No, it's wrong. It's a wrong answer. And you might have picked it if you just saw protein synthesis. So our next choice, the damaged rough ER and low ribosomal RNA levels suggest that protein synthesis is impaired because the rough ER is the site of ribosomal attachment and protein synthesis. This is probably our right answer. The malfunction of the mitochondria has caused decreased ATP, which would affect protein synthesis. Now, if we did have a malfunction in mitochondria that caused decreased ATP production, might we have a decrease in protein synthesis? Yes. But is that what is happening here? No. It literally told us it's an issue with ribosomal RNA levels in the rough ER, so that's not our answer. Reduced activity of the lysosomes is leading to an accumulation of undigested proteins, which impact overall protein synthesis. It's not what's happening here. Right? We are having a decreased ribosome availability. All right, our next concept, trophozoites and cysts, of course. Trophozoites, in chapter 5, trophozoites are the active, free-living, replicating, multiplying equivalent to vegetative. Cysts are dormant and inert, but they're also very hardy and resistant. Hardy, resistant. These are stages of the protozoa. Not all protozoa make cysts, but all protozoa, when they're active and multiplying, we call them trophozoites. Okay. All right. So just FYI. And again, there's a lot more detail here if you need a little bit further review on these concepts. So we're going to move on to our questions. A parasitology lab, hold on real quick. A parasitology lab is examining a patient's stool sample and identifies both trophozoites and cysts of a protozoan. So we see both. There's trophozoites, which are active, and cysts, which are dormant. And I haven't looked at the question yet. I'm already making notes. The patient is experiencing GI symptoms, such as diarrhea and abdominal pain. So there's an active infection here. That I don't know what happened there, but it's infection. We're actually having infection and disease here. Now, there's a difference between these words. Remember, infection is when a microbe is growing in you. Disease is whenever your health is deviating from it. So you can have infection without disease, and you can have disease without infection, like diabetes, for example. Okay. All right, so back to this. We have infection and disease going on. Based on the presence of both of these life stages, which of the following statements best explains their roles in the infection cycle? So let's look at our choices here. We know trophozoites are active. We know cysts are dormant. How did they each play a role in this infection in our patient? Trophozoites are responsible for causing symptoms in the host due to their active feeding, while cysts help in transmitting the protozoan to new hosts through environmental contamination. So this sounds accurate, but I want to see what other choices are. Do we have any better explanations? Trophozoites are primarily involved in survival and transmission, whereas cysts are involved in causing the infection within the host. So what we need to understand a little bit here, are cysts causing infection? They can't because they're dormant. Cysts do not multiply. Infection means that the organism is multiplying in you. Trophozoites multiply and cause infection. The infected growing organism is what's causing our signs and symptoms. It's producing toxins, it's growing, it's eating off of our tissues and things like that. So all of that is going to be from our trophozoites. So B is wrong. All right. And also the survival form, the one that is hardy and resistant, they do that to survive. So that would be the cyst. So these choices are kind of mixed up on B. C, trophozoites and cysts both contribute equally to infection, symptoms, and transmission of the protozoan by actively feeding and surviving harsh conditions. This is wrong. Do they equally contribute to the progression of the disease? Yes. But do they equally contribute to the infection and the toxins and all that? No. The trophozoite does the infection, but the transmission, how the organism got in your body, was because somehow the cyst got in, became the trophozoite, which then started multiplying. Okay. D, trophozoites are dormant. No, the cysts are, so this is wrong. D isn't our choice. A is still our best choice here. Okay. In an experimental study, researchers exposed protozoan cysts, dormant and resistant, to various environmental stressors such as heat and desiccation. If you're a good, proper cyst, you should be resisting those things. The study finds that the cysts survive these conditions, while as the trophozoites, which are not hardy, not dormant, but they are active and free living and multiplying. What can be concluded about the function and adaptation of protozoan cysts? Are cysts adapted for active growth? No, that's trophozoites. Cysts are, and listen, did I read the whole choice on that one? No, I came to one part that was incorrect. I don't need to read the rest of it. If one part of it is correct, if one part of it is incorrect, the whole thing is not the right choice. That's true, okay? If you get to a part that's part of it is correct, you need to be sure all of it is correct for it to be correct, okay? Cysts are designed for survival under adverse environmental conditions. This part's true. Maybe it's this one. Making them highly resistant to heat and desiccation. Yes, whereas trophozoites are less resilient. All of that seems true to me. It's probably B, but let's go through our other choices. Cysts are more susceptible to environmental stressors compared to trophozoites, which are more durable. This is switched. It's not C. D, cysts are involved in active reproduction. Again, these are switched. Just a different wording of switching them. B is still our right answer. All right, next we're on to some chapter 7 concepts, our viruses and our prions. Cytopathic effects. So we did talk about these. Cytopathic effects are things that we can see actually changing about the cell as a result of the viral infection. This would include things like the cell swelling, the cell rounding. We have detachment of the cell to surfaces or to tissues. We have cells bursting open potentially. We have syncytia being formed. Again, whenever cells merge together as a result of the viral proteins causing accidental docking with our neighboring cells. And then inclusion bodies, when we're building up a ton of viruses inside of our cells. This causes buildup of the proteins and the genomes of those viruses that can be visible inside of our cells on our microscopes. And so these are called inclusion bodies. So all of these are effects that we can see happening to the cells as a result of a viral infection that we might be able to see even though we can't see the virus itself. So let's get into some questions about our cytopathic effects. Which of the following best describes a cytopathic effect in the context of a viral infection? So before I answer it, I'm just thinking of things that happen to the cell that we can see in microscopes caused from a viral infection. All of that is key. So let's look at our choices. A viral protein that inhibits host cell replication. Can you, if you look at your patient sample, actually tell if the cells are no longer replicating? No, you're just going to see cells sitting there in a microscope. Can you tell if they're replicating or not? It takes days for you to be able to see that. This is not realistic. A structural and functional change observed in host cells due to viral infection. Yes, these are things that you might be able to see in the cells as a result of the viral infection. The process by which viruses are released from infected cells. With a normal microscope, you wouldn't be able to necessarily see that viruses are going through exocytosis or that they're budding out. This stuff is happening at such a microscopic level, you wouldn't be able to see that in a normal microscope like we use in our lab currently. This stuff wouldn't be able to be seen with that. The ability of viruses to integrate into the host cell genome. Can you see that in a normal microscope? No. You would be able to see structural and functional changes potentially caused this. Again, syncytia. We can see that the cells are merging together. We can see that they're not attaching to surfaces anymore. We can see that they're rounding up. We can see those sorts of things. A laboratory technician observes that cells in a culture have fused together to form giant multinucleated giant cells, and they exhibit swelling as well. So we've got swelling, and we've got these fused cells, fused. And remember, when cells fuse together, we call that syncytia. Which of the following viral infections is most likely associated with these cytopathic effects? I could have just left it that alone and left it there if this was Unit 4, but we're not anywhere near that yet. So now I give you a little bit of information to help you out based on the name alone. Based on the name alone, I can see one in here that has something to do with these terms in it, and that's D. The other one's just based on the name. Might they? Potentially, maybe influenza virus causes syncytia or swelling or both. But based on the name alone, it's RSV here, the syncytial aspect. Okay. This is basically just asking you to recognize that the term, this is a fancy way of me saying that syncytia, what does that mean? Just a fancy way of doing that. Okay. Don't read too much into that. All right, next, chapter 7, the temperate phages, the lytic cycle, the lysogenic cycle, and lysogenic conversion. This is a little bit more explanation of those things. So you can see if you need to read up a little bit more on those phases there. I'm not going to re-explanation all of this based on this chart here because I want to draw a little bit of a kind of cheat sheet over on this other one as my little review of it before we go on. Okay. All right, so again, a temperate phage can switch between the two. Lytic, we're making new viruses that burst out of the cell. Lysogenic, we're incorporating the prophage into our genome. Into the bacterial genome. Sometimes, I'm trying to write it sideways. Sometimes that can cause new traits. And that is what we call lysogenic conversion. And like I said previously, that process, watch this one. This whole process is called transduction. That's not going to be the question, but I just want to remind you guys of that. Some of these processes and terms and content that I repeat a lot, the reason I'm repeating it a lot might be because I'm going to test you on it, of course, but it also might be because you need to understand this in order to understand future chapters. Like that's why I've repeated a lot, okay? Hint, hint, chapter 8 is going to talk about transduction some, okay? Because we can use it as a tool. All right, I have to hurry up because I have a meeting. Okay, so first question, which of the following best describes the main difference between lytic and lysogenic cycles of temperate phages? The lytic cycle, again, new viruses. Lysogenic cycle, again, incorporating it into the genome. So our choice A, the lytic cycle involves the integration of the phage DNA. That's already wrong. That's the lysogenic. Question B, the lytic cycle results in destruction of the host cell through lysis. Now, I said new viruses burst out. Bursting out is called lysis. So this part is correct. While the lysogenic cycle involves the integration of the phage DNA into the host cell genome without killing the cell right away. Lysis obviously would kill the cell. Again, this is bacterial. It always happens with bursting out of the cell and killing the cell. This seems like it's right. C, the lytic cycle occurs in all types of bacteria, while the lysogenic cycle only occurs in gram-positive. That's where it went wild. Okay, no, not just gram-positives. We didn't say anything about gram-positive versus gram-negative with any of these phages. So that's not happening. The lysogenic cycle produces new phage particles immediately, whereas the lytic cycle does not produce phages. These are switched. So B is our answer. Our next question. A patient presents with a bacterial infection caused by a strain of Corynebacterium diphtheriae. This is a bacteria. The lab results show that the strain, this bacteria, is producing toxin called diphtheria toxin. Based on what we have learned about lysogeny, of the following, which of the following explains why this strain can produce the toxin? This is suggesting, and again, if this is an exam question, I would give you a little bit more information about this. But based on what we learned, this sounds like lysogenic conversion. That a virus gave this bacteria the toxin genes. So is this suggesting the bacterial strain is carrying a prophage that is integrated into its genome, which encodes the toxin? Well, that immediately got off the bat. It's probably the correct one. Is it suggesting the bacterium has a high mutation rate that randomly was able to make toxins? It's not suggesting that based on what we're learning here. Bacteria has high resistance to antibiotics. This is completely unrelated to the toxin. Not that. The infection has caused the bacteria to undergo genetic recombination, leading to toxin production. This is kind of what is happening. It is kind of doing that, but the more correct answer here is A, are we changing the genetics of our bacteria by this prophage doing this? Technically, yes, but the more accurate description is going to be A. Okay, and like I said, the exam questions are going to be much more clear on what it's asking you. And if you're ever taking my exam with me in person, you can always ask me clarifying questions on what the question is asking. Or if there's a term you don't understand that definitely wasn't a vocab term, or you want a little bit more reference to understand something a little more, you can always ask for clarification. So that is it for chapters 5 and 7. Do you guys have any questions for me? If not, that's fine. And I will post this as well as an older video for people online for you guys to study in the future. I've got to get to a meeting in 10, I've got 10 minutes to get to a meeting. And so email me if you guys do have any questions. I hope this was helpful. Happy studying, and I will see you for your exams next week. Remember, I'm keeping my review at 4 p.m. on Sunday via Zoom. So I hope you guys can join me for that. And I'll see you then. Thanks, guys.
Thank you.
Yep.